Okay, top and bottom one stayed liquid, and the one in the middle, staph aureus, turned solid. So why did that one turn solid, guys? Well, it turned solid because staph aureus produces coagulase. Do you guys know what coagulation is? That's when blood clots, isn't it? That's when it clumps up. So when I set up the plasma tubes, that's the only one that clotted because it produces the enzyme. Does this make sense to everybody? Excellent. Okay, so today we're going to set this up, and I've decided to have mercy on you today. And I'm going to have this side of the room set up the plates for the PEA and EMB. And I'm going to have you guys on this side inoculate these tubes here. Sound good? And then we'll take a look at what these results are next time. So let's go back to page 44. I want to explain a couple of things with the PEA and EMB. So PEA stands for phenyl ethyl alcohol, and it inhibits DNA synthesis in gram-negative bacteria. So it inhibits DNA synthesis in gram-negative. I'd like you guys to think about this. We said gram-negative has an outer membrane, right guys? What's a membrane made of? Lipids. So this alcohol then, phenyl ethyl alcohol, can slip past and then it interferes with the DNA synthesis of gram-negative. Okay? So gram-positive are going to be okay, they can grow just fine. Gram-negative will be inhibited. For the EMB plates, stands for eosin, methylene blue. Inhibits gram-positive growth. So you will inhibit then the growth of gram-positive. So these are really a good way to double check your gram stain, isn't it? It's another way to verify whether you have gram-negative or positive. So can we say that they select for coliforms? So that's a good term to know, coliforms. And a coliform class is a gram-negative facultative anaerobic rod. All of that makes sense now, right? You know what gram-negative is? Facultative anaerobes can grow with or without oxygen, right? And a rod shape, we all know what that looks like. So, excellent. And 3 results can occur, guys, based on fermentation. We can have, and this is really cool, guys, the metallic green. If you have a strong fermenter, a pink to purple color, and that would be if you have a low to moderate fermenter. And the last type, guys, no color. And that would be a non-fermenter. Okay, so here's what we're going to do. For all of these guys today, I'm going to ask you, please, we've been saying all semester you have to label, right? So your initials should be on there. Each of you has a different type of bacteria, so again, you guys are going to be setting up the plates. Okay? Today's date, and it's going to be 37 degrees C. So, you'll label an NA plate. So we need to verify, class, that your bacteria is alive. So you're going to label an NA plate, a PEA plate, and an EMB plate. Then you're going to aseptically quadrant streak. With your bacteria. And then incubate at 37 degrees C. So, quadrant streak means how many directions? 4 directions, right? So, heat flame your lip, cool it, dip it in your bacteria, go in 4 directions for each one of those plates. So that's going to be your guys' job over there. Are you up to the task? I thought so. Excellent. Okay, now, starting on page 51, we're going to get into a lot of other types of biochemical tests. So I want to make a statement right now. You're going to have a project for an unknown that's going to be worth 50 points. You're going to have a big lab report to write. On page 50, so please write this down. On page 50 is a good general background information, on biochemical tests. This will help you with understanding what you're going to be doing the next 2 weeks. Also, you have to write an introduction to your lab report, and you can use some of that general information in your introduction if you'd like. Sound good? Okay. Now, another thing I know you worry about, because you guys always worry a month in advance about exams, am I right? Okay, I knew it, I could tell. So, for a lot of what you're going to be doing this week and finishing up next week, guys, you want to know what you're testing for and what positive results are. So for instance, you're testing here to see if DNA synthesis is inhibited or not. Here, you're testing for these coliforms, correct? Here's some of the results: green metallic sheen, pink to purple, etc. So let's talk about the PR fermentation and what you're testing for. Well, in the name here, guys, we're testing for fermentation. Okay? So testing for fermentation, and yellow is positive. And the reason for that, class, and again, if you watch that video, it's because the phenol red will turn yellow if an acid is produced. So if a sugar is fermented, guys, acid gets produced. Does this make sense? Another really cool thing. If gas is produced, you will have a bubble in the Durham tube. So this is really cool. So again, this side of the room over here is going to set up 3 sugar tubes for this. When you look inside, guys, really cool. There's another tiny tube on the inside to collect gas. How many of you have ever baked bread? Anybody ever baked bread? And you know when it's rising, do you guys see like the little bubbles that come up? That's fermentation. And that's from gas that's being produced. So, what you guys are going to do over here is you're going to label 3 PR fermentation 2. And just so you know, we have one tube that has glucose, one has sucrose, and one has lactose. So you use a loop for this. Is that clear, guys? Use a loop for this. Stick it in, shake it off, E-flame your loop again, go in, etc. For that, so aseptically, inoculate, and then we're going to incubate at 37 degrees. Okay. Now the next one is also on that page, guys, and we have one tube that is going to be split into 2 next time after it grows to test for 2 different things. Okay? So the tube that says MRVP is for the following. Methyl red, that's the MR, and this tests for mixed acid fermentation, mixed acid fermentation, and red is positive. Everybody got that? So that's the MR part. For the VP part, Vogt cross power. So for this one, tests for acetone. Test for the chemical called acetone. And if you guys look in your lab book, it explains how you can go from pyruvic acid to acetone. You can also go from pyruvic acid to other stronger types of acids. And kind of a reddish brown is positive. So what you're going to do today, guys, is label 1 MRVP broth and aseptically inoculate your bacteria, and then we're going to incubate this also at 37. So, just so you guys are aware, next time, we're going to split and add reagents. Okay? So one of the things, again, please read the information in that handout and in your lab manual, guys, because for some of these you have to add reagents. Some of them, like this, and this one, they're going to do their thing without adding anything additional. But the MRVP and the PAD, you're going to have to add reagents to. Is that clear, everybody? Okay, so the last two, again, that you guys will set up over here are on page 52. And I want you to know an equation. Oh, no, chemistry. Yes, chemistry. So, This tube, guys, we're going to start out with a urea tube, urea agar tube, that's kind of an orangey color. Okay? Now, if the enzyme urease is produced, you're going to get ammonia plus carbon dioxide. And a beautiful bright pink is positive. Look at the picture atlas. It's got all of these results. You're going to need those for your unknown. All right, guys? Now, you're going to label your tube. And use the needle. So this is the only one, guys, that you're going to be using the needle. So you guys know in your metal cup there, you're going to use the lid for everything else, but you're going to use this instead. So when you guys go in the tube, guys, be careful. Just rest this on top of the bacteria. Rub it a little bit back and forth, take it out, and stab it right in. Get those frustrations out, okay? So when you do that, you use the needle and inoculate urea agar. Let's say here's your tube. Stab it about halfway down, okay? So stab it about halfway down. If any of you over here feel left out and want to stab one, you are more than welcome to stab or inoculate any of these other ones. But if you want to stab one, seriously, just for the experience, go ahead and grab one. Sound good? Okay? And then again, we're going to incubate at 37 degrees C. And then our last one, guys, is also on that same page, and that's the phenylalanine deaminase. So what happens, guys, is how many of you have heard of phenylalanine? It happens to be an amino acid. Any of you ever heard of PKU? Okay, that has phenylalanine in it. So anyway, what happens is the amino acid phenylalanine can be deaminated to produce pyruvate acid. Pyruvic acid, sound familiar? Pyruvate, right, guys? So if we take this phenyl off, what are we left with? Pyruvic acid. So that's the reaction that occurs. So what you're going to do for this one is label 1 PAD slant. Now, when I say slant versus just like an agar tube, I mean, it's what you guys have been giving you, right? Like I've been giving you bacteria that are like on that slant. So you're going to inoculate this one by doing what we call a fishtail, okay? So aseptically, fishtail streak PAD slant. So when you do this, you take your loop, guys, go to the bottom of the slant, okay? And you're going to zigzag it going up. So basically that, you zigzag it going up, and then incubate at 37 degrees C. And then next time, you're going to add iron chloride. Okay? Are there any questions, guys, about this? As I said, you guys will do the Na, the MBPEA. You guys will do these. However, if any of you want to do more than just what you have to, I'm fine with it. Like I said, if you guys want to practice doing a stab, might be the only chance you're going to get. That's up to you. Right? Have fun, guys. All your tubes and such and plates are over there.